My laboratory became interested in this disease, vacuolar tauopathy, about 4 years ago when I was asked to do a brain autopsy on an individual with frontotemporal dementia. And in this case, um, the person who donated their brain for research also had two siblings who were affected by FTD and a parent. And so there seemed to be a strong genetic component or hereditary component to this disease. And when I looked under the microscope at the brain tissue in this one case, in the regions of the frontal lobe that were degenerating, we saw a buildup of protein called tau. So tau is a protein that can aggregate inside neurons. In this case, we did see these protein aggregates in the form of what's called a neurofibrillary tangle. And these tangles were similar in how they appeared and the proteins that made up the tangle to the neurofibrillary tangles that we see in Alzheimer's disease. And so it's the same protein and the same structure that appears to form these tau aggregates in vacuolar tauopathy and Alzheimer's disease. In other areas of the brain, we also saw abnormal vacuoles. These vacuoles look like empty holes that were forming inside neurons. And because we saw this, we gave this disease the name vacuolar tauopathy. It's a pattern of pathology that has not been described before, and so we thought that this was very interesting to pursue. Now, with the help of our colleagues, including Viviana Van Gerland here at Penn and the NIH Undiagnosed Disease Network, we both found that this individual had a mutation in a gene called VCP. So VCP encodes for a protein, the volosin-containing protein, and VCP's job in the normal setting is to bind to various complexes inside of a cell and helps pull apart those complexes. So if you imagine that life is a series of biochemical reactions where proteins and lipids and nucleic acids come together to perform certain reactions, you also need mechanisms to be able to pull apart those complexes, and that's often what VCP is doing. And so this mutation we discovered in VCP when we tested in our laboratory turned out to be a partial loss of function of VCP activity. What does that mean? So it means that VCP usually takes energy to break apart these protein complexes, and the mutation made the VCP protein less able to do so. We next asked, how does this relate to the buildup of these tau protein aggregates in neurofibrillary tangles? And so what we found was that VCP seems to be able to bind to these tau aggregates themselves and help pull them apart. And so VCP seems to be acting as a tau disaggregase, meaning it can bind to and help pull apart these tau aggregates. So if you have a loss of function of this protein, that means the tau aggregates build up and you form these neurofibrillary tangles. So we think this is a new function for VCP within a cell and is linked to this new disease called vacuolar tauopathy. Now, this mutation in this disease appears to be extremely rare. While we were working on our studies, we discovered that there's another family in Greece with the same mutation who also appears to have vacuolar tauopathy. But other than that, this disease hasn't really been seen yet in many families, and so it appears to be extremely rare. And so what's the relevance of this to more common disease such as Alzheimer's disease? And so what I'll say is that rare genetic forms of disease often allow us to have insights into the mechanisms that cause more prevalent common sporadic diseases or non-genetic diseases. So for example, in Alzheimer's disease, there are mutations linked to the production of a protein called A beta, amyloid beta, and this protein builds up as amyloid plaques in Alzheimer's disease. And so it's no surprise that now a major focus of therapy development for Alzheimer's disease is to try to target these amyloid beta plaques. Similarly, we think there's a possibility to target VCP. So if a loss of VCP function results in the formation of neurofibrillary tangles, perhaps the converse is true, meaning if we can find a way to boost the activity of VCP inside of a cell, that might help break apart tau aggregates. So VCP might be a therapeutic target, meaning if we target VCP to try to boost its activity inside of a cell, that might help promote disaggregation of tau, and may be relevant not just for this rare genetic form of disease, but for more common sporadic diseases like Alzheimer's disease. Again, the same neurofibrillary tangles are building up in this rare genetic disease as is seen in Alzheimer's disease. And so perhaps targeting VCP is a way to help rid the cell of these tau aggregates in the setting of Alzheimer's disease.